Swayam Prabha. Digital India. Educated India.
Hello all, welcome to Astrochemistry. This is lecture number 83. Now in the previous lecture, we were discussing about how the UV Miller experiment explained the formation of a lot of organic molecules which are relevant for the evolution of life. That is, a lot of bioorganically relevant molecules. We were looking at the broad mechanism of the formation of some of these basic entities of life, and we started off by looking at the mechanism for the formation of HCl. Now, one important question, which should come to you at this stage is, recall to you, what do you think were the starting materials that were taken in the UV Miller experiment? What did I say? In the UV Miller experiment, what were the starting materials did we take? Well, we took hydrogen, ammonia, and methane. If you notice that I did not tell you anything about water, correct? Now, why was it? Well, it makes sense because, this came about because of UV's hypothesis that, the early Earth's atmosphere possibly mimic that of Jupiter, and there is no water on Jupiter. However, on Earth, especially if you consider putative early Earth type conditions, there should definitely be some water arising as a result of either volcanic activities or, as a result of some kind of a cometary influence or a meteor getting dropped, etc., etc. Okay, so note that even though these were the molecules taken to represent possible early so even though these were the molecules taken to represent the possible early Earth atmosphere, right? Water was also taken in a separate vessel. In their experiment, that is in the UV Miller experiment, right? Water was also taken in a separate vessel as it represented, the scenario due to volcanic activity, etc., etc. Okay, represented the scenario due to volcanic So what I've written is water was also taken because water could have been formed. Okay, the scenario I'm talking about is the formation of water, as a lot of volcano erupts. Why should the volcanoes erupt? Recall we were discussing about our cold and hot accretion theories. In any case, right? In any case, due to the iron getting sunk, a lot of smaller molecules would be coming to the surface, right? And definitely it is very likely that water is also one of them. So therefore, as part of representing the early Earth atmosphere, these molecules were taken. As part of representing the volcanic activity, water was also taken. Please note this point. So these three were not the only molecules to be taken. This was also taken. Water was also taken, but for separate reasons. Okay, it should not be understood that water was also taken as part of representing the Earth's early atmosphere. No, this is to when I say atmosphere, I'm talking about the gaseous mixture that is already part of a light envelope. All right, water is taken to represent the internal structure, the internal structure which creates a source of the oxygen atoms. Because if you think about it, right, see, the constituents of life, right, the molecules which are responsible for life are, you can think about them in terms of the peptide bond. Okay, the amino acids contain the peptide bond. So what is the peptide bond? It is the C double bond O NH bond, right? So you have H, you have C, you have N. But what about O? You need a reservoir for O, right? You need a starting point for oxygen, and water presents that beautifully. All right, so please note that. And yet again, I will tell you that the water was not taken, right, as a representative of early Earth atmosphere as being an analog of Jupiter's. All right, it was taken as part of understanding the not understanding, sorry, mimicking the volcanic scenario, right, that was happening on Earth. So overall, along with these three molecules, water was also taken in a separate chamber. And again, these are the reasons are interpretations. Okay, UA and Miller did not precisely tell the exact reasons. They were not clear in the sense that they did not tell that, oh, it is to represent the Jupiter-like atmosphere that we took these three, and it is to get the volcanic type scenarios that we take water. All right. It is understood overall that we also need oxygen atom, and water is a great supplier of oxygen atom, and especially given the fact that your water can easily be arising as a result of volcanic eruptions. Okay. All right. So on that note, I'll keep this intact. I'll delete this later. We shall look at the chemistry of water in the context of the UV Miller experiments. Okay. So from water, the following reactions can happen. That is, once again, your water, when you subject it to some sort of an electric discharge, you get, this is also mimicking the photodissociation scenario. So you shall be getting your OH radical and H radical. All right. So see, they were in fact taking hot water, okay, boiling hot water. So therefore, there is this phase transformation between the liquid phase to the gas phase, and one could imagine this reaction is happening in the gas phase. Okay, right? Because you supply enough energy to first heat the liquid water, and then once you keep supplying energy, the bond breaks. So you get like OH dot and H dot. All right. Okay, so this is understood as happening in the gas phase, even though you start off with liquid water in the experiments. All right. So your methane is also part of the UV Miller starting mixture, right? So methane reacts with the OH radical. So you first generate the methyl radical, and you form water again. See, akin to what happened in the second step of the HCN formation, you kind of start a catalytic cycle. All right. So you have the methyl radical generation, and from the methyl radical, then the methyl radical reacts with the OH radical form to actually form methanol in the gas phase. And once methanol is formed, well, methanol, once you subject to photolytic oxidation, right, what happens is it gives rise to formaldehyde and hydrogen. Now what is this? This is HCN and this is formaldehyde. Now why were we particularly concerned about the formation of these two small molecules? Well, the magic is about to happen. So starting with these two molecules, so starting with formaldehyde and HCN, they further react. Okay, so you have H plus HCN, right? What happens is that, and there's also some ammonia, right? Now all of these three react in some specific sequence, right, to give you this product, to give you your NH2CH2CN. And this, upon hydrolysis, gives you, and this is nothing but glycine, and this represents the most fundamental peptide linkage. Please note this. Okay, right? So I have not written the overall mechanism. You can also write the reaction mechanism for this. First, what happens is your HCO and ammonia react, and then like to the adduct, HCN is added. I've just written the overall reaction. Remember, I think in lecture 71 I've told you a chemical reaction does not proceed in a single step the way we have written it, right? There are lots of sub steps as part of the mechanism which result in the overall final equation. Okay, please note that point. So I'm not going into the exact mechanism. In fact, this is what is called as the Strecker synthesis of amino acids. The Strecker synthesis of amino acids explains how amino acids can be formed from a simple mixture of HCN and HCHO, which can in turn be formed from the starting materials taken in the UV Miller experiment, along with water also. All right, are we good? So therefore, what have we done so far? What we have done is we've seen a plausible pathway to result in amino acids, which are the fundamental units of proteins and a lot of life forms, right? Starting from very simple molecules. Now, indeed, why should I be explaining this? I'm explaining this because glycine and I think 4 or 5 other amino acids were actually obtained in the 1950s when UV and Miller performed their experiment. Okay, so note glycine and other amino acids. If I'm correct, 4 or 5 more. Okay, 4 more, including glycine, were formed in UV Miller experiments. Now, what is the overarching conclusion? The overarching conclusion is that starting from very simple starting material, subject to intense electric discharge, which is a mimic for the radiation during the possible early Earth conditions, you can form the molecules which are eventually going to lead to life. Please understand this clearly, okay? So I'll just sum up what we've seen here. In fact, this was the scenario before summing up. A small digression. See this, I told you that 4 or 5 amino acids were formed in 1950s. When the same samples were, I think I alluded to this briefly in the previous lecture also, when the same samples were reanalyzed, probably some 40-50 years later, somewhere in the late 2000s or early 2011 or 12 around that time, it was found that a lot more amino acids, more than 5, some close to 17 or 18, almost all of them barring the sulfur containing ones, were obtained via the UV Miller process. So all this data indicates that under the right conditions, complexity, I'll say chemical complexity can emerge from a very simple, when I say simple, I'm talking about in terms of the number of molecules, the chemical structure, etc., from a very simple mixture, which implies that, the molecules responsible for life we're talking about the various peptides, the carbohydrates, all of them. I'll give some more examples of molecules, which will make this point even more emphatic. Okay, so the molecules responsible for life could have originated via simple chemical processes. On Earth itself. In other words, you have some deposition of a mixture of molecules via meteorites and cometary collisions. But not everything needs to be, coming from the outer space, that is the interstellar medium, or via collision by meteorites and asteroids. What can instead happen is some stock, some once you have a fundamental stockpile of like very small molecules, they can further react to give rise to chemical complexity on Earth itself. So this is what the UV Miller experiment demonstrated. Note a big caveat, I said right conditions. How do we know for a fact that the starting conditions that UV Miller took was indeed appropriate? Well, there is no definitive answer to that yet. All right. It is in fact a very strong point of debate in the prebiotic chemistry and astrobiology community as to whether the mixture to be taken in the beginning, should it contain some carbon dioxide or not? That is, it basically refers to whether the atmosphere is reducing or oxidizing. So lots of theories are proposed for that, but those are research topics. Okay, just understand that at an undergraduate level to begin with, small chemicals can give rise to complex reactions. I haven't gone through the mechanisms of each of the individual steps. That can also be done, but in the interest of time, I've skipped them. All right. So now what we'll do is we shall look into how some of the other fundamental, moieties responsible for life, how they are generated. Okay. So the next moiety that we shall be looking into is how are the carbohydrates generated? Well, I'm not going to give you the complete sequence. I'm just going to tell you that. By the way, I've already told you this in this course. Do you recall when? I'm talking about probably in the very first or second lectures when I was talking about a glycoaldehyde, the ketoenol tautomerism, etc. Do you remember? Okay, so this has already been covered to some extent. All right. So how are the carbohydrates obtained? The answer is you start off with formaldehyde, right? And formaldehyde, we already saw how it is formed from water and methane, right? Methanol is obtained and methanol gets oxidized to get formaldehyde. So formaldehyde, if it polymerizes, you shall be getting HCHO n times, which is nothing but the carbohydrates. The key point to note here is that, the process of HCHO, going to HCHO twice, glycoaldehyde, right? This process alone is extremely difficult, But once, in the second, the dimer, that is like glycoaldehyde is formed, from here onwards, we can go all the way up to n equal to 6 readily. So note this point, okay? So 5 or 6. But there is still a caveat. The caveat is that if you have seen the structure of DNA, what is the sugar base for DNA? It contains a sugar called as ribose. One caveat, okay, right, to the sequence is that this process called as the Formose reaction, okay, does not yield enough ribose. So how exactly ribose, which is a 5-membered sugar, it does go all the way to n equals 6. So the n equals 5 sugars are also formed. So this specific sugar called as ribose, this process does not yield that. Okay. So how exactly is ribose formed is still a mystery. Okay, right? So ribose formation still debated. All right, so this is as far as the carbohydrates are concerned, the carbohydrates and the sugar moieties. The next point is, what about how are the nucleic acid bases obtained? Well, what are the nucleic acid bases I'm talking about? I'm talking about i.e. adenine, thymine, guanine, and cytosine. also along with uracil, which is also important. How are these formed? If you think about it, if you especially consider adenine, right? So your adenine, I'll let you write the structures as homework, okay? But I'll just give you the molecular formula for adenine. If I'm correct, it is C5H5N. N5, sorry. Yeah, so C5H5N5. Can you already spot the connection between this molecule and a molecule we've already seen in this lecture and the previous one? This one. Similar to how the Formose reaction occurs as a polymerization process. Well, you understand that this could have definitely come as a result of the polymerization of your HCN, right? So this is nothing but HCN 5 times. So somehow, 5 HCN molecules come together, and that results in the formation of adenine. And adenine can react to give rise to other variations. So similarly, there are other mechanisms proposed for the formation of guanine, cytosine, and thymine also, and uracil also, which is formed by one reaction from cytosine. Are we clear? So therefore, you can also explain broadly the nucleic acid bases. Although I've only given you one example, in the interest of time, you can always study the other three also. Now, the next thing is, your DNA contains, 4 basic moieties. The sugar, which is ribose, about which we saw, it is of course obtained as part of the Formose reaction from formaldehyde, but the yield is pretty low via this mechanism, so other mechanisms are also important. I'm not going to go to the details because it's an active area of research. Similarly, the base can be obtained by polymerization of HCN and related small molecules. There is one more thing. It also contains the phosphate linkage, not 4 but 3, I'm sorry. Okay, sugar base and the phosphate linkage is said to be, coming from the minerals which are part of meteorites. Okay, so phosphate linkage proposed to come from especially some iron, nickel, phosphorus containing minerals. So I think they're called as Schreibersite, SCHREIBERSITE. Okay, some Schreibersite minerals which have been detected in the meteorites. So the meteorites can give rise to the materials which eventually constitute DNA. Okay, now there is a huge debate about what came first. Did the DNA come first, or did the RNA come first? And in what sequence were these molecules assembled? And all of these remain active areas of research. For our purpose, all we need to understand is that the larger molecules, such as the DNAs, etc., there can be the primitive blocks of the larger molecules, such as DNA, can be readily formed under an early Earth type condition. So how did they actually assemble? What about the form? What about the function? When did they emerge? What about RNA, DNA? All of this is going to be a completely a new course, in fact, okay, a new course which begins from here, titled Astrobiology. Okay, right? So it's an active area of research where people are pursuing how this mixture, right, led to DNA, and whether it is DNA which came first or whether it is the RNA where that came first, and what about some other molecules, related molecules such as peptide, nucleic acids, etc., etc. So we're not going to go there. I'm just going to tell you that even on Earth, under proposed early Earth conditions, from a very simple scenario involving very few molecules which are representative of the reducing atmosphere, along with water, we can actually obtain the sugars, the nucleic acid bases, and also the the phosphates also, right? So we can obtain all of this, and in some combination, eventually, the polymeric molecules, the biopolymers such as DNA, RNA, they may have formed, and they lead to life. Now, before I end this lecture, I want to talk to you about just one. I want to tell you about like one important property of water. Okay, the significance of water, as the basis for life itself. And why is water special? Hey, because water, especially liquid water, is only found on Earth. Okay. Now, for this, I'll go back to so as part of the next topic, I'll go back to the surface temperature and pressure on Earth. So the surface temperature on Earth was roughly 285 Kelvin, right? And the pressure was around 1 atmosphere. Now, based on the phase diagram, this implies that water could exist in the liquid phase on Earth. Now, one may think that, of course, this is an obvious fact. I mean, we see liquid water around us all the time. So why is it important from the context of life on Earth? Okay, this is in fact extremely important. Normally, 4 reasons are listed as to why this is an extremely important point. The 4 reasons are that the first one is that so water has a 100-degree range where it could be liquid. Right? Of course, predominantly liquid. The other forms I'm talking about at 1 atmosphere pressure, which implies that water supports different forms of life, some of which are resistant to high temperatures, some of which are resistant to low temperatures, and some of which are active at ambient temperatures, which implies that the range of resistance temperature is massive, is amenable, sorry, is useful, I would say, is useful to generate various forms of life and structures. Not just life, it need not just be life, it can also be associated with a lot of chemical structures and assembly of them. So this is one point. The next point is that this is very, very important. Density of ice being less than density of water at around until 4 degrees. This implies that aquatic life, which is extremely critical for survival of humanity, right, can exist. Okay, this implies that aquatic life can exist without the species freezing, just getting completely frozen by ice layer, without the life forms not getting frozen. So what I've written here is, because the density of ice is less than the density of water, right, until about 4 degrees, even at low temperatures, several of the species can survive. Not everything will get frozen. Only the top layer, right, ice will float atop water. So therefore, you'll have like life forms beneath the sheet of ice in water surviving. So this is yet another important point. I'll continue with the importance of water in the next lecture, and I'll also talk to you about the significance of a very cool idea in prebiotic chemistry and also astrochemistry, in fact, about chirality in the next lecture. So to sum up what we saw in this lecture, we were discussing about the need for water also in the UV Miller experiments and why that was also added. And after that, we were discussing the mechanisms of how various molecules relevant to biology were formed. And then I started telling you about the significance of the 280, the 285 and degrees and 1 atmosphere pressure, as the surface pressure, the surface temperature and pressure for Earth, and why that is important for water, right, to exist as a liquid, and how that in turn, the properties of water, how they in turn make Earth a unique place for life. Okay, so on that note, I'll see you in the next lecture. Thanks a lot.